For a long time, classifying life seemed like a matter of sorting organisms by appearance. Animals with fins went in one mental box. Animals with feathers went in another. Worm-like creatures, fish-like creatures, reptile-like creatures, and mammal-like creatures all seemed easy to recognize from the outside. Cladistics changes the entire question. Instead of asking which organisms look similar, it asks which organisms share the most recent common ancestors. That turns classification into a map of descent. A group is not considered natural just because its members resemble each other or live in similar ways. It is natural only if it represents a real branch in evolutionary history. This is why cladistics became so important in modern biology. It forces names and categories to follow the branching pattern of evolution, even when that pattern conflicts with familiar everyday labels. The basic unit of cladistics is the clade. A clade is an ancestor and all of its descendants. If you picture an evolutionary tree, a clade is what you get when you choose a branching point and include everything that grows from it. The size of the group does not matter. A clade can be enormous, containing many major lineages, or it can be small and recent. What matters is completeness. If a group includes the ancestor but leaves out some descendants, it is not a proper clade. This makes cladistics stricter than many older classification systems. It does not allow scientists to keep a convenient group if that group cuts across the actual family tree. Every valid clade is therefore a claim about ancestry. These organisms belong together because they descend from the same branch. To decide where branches belong, cladistics looks for shared derived characteristics, also called synapomorphies. These are traits that appeared in a lineage and were passed down to its descendants. The word derived is important. A trait is most useful when it marks a newer branch, not when it is simply an ancient feature inherited by many distant relatives. Scientists are not just counting similarities. They are asking which similarities reveal common descent. That distinction matters because evolution can be deceptive. Unrelated organisms may independently evolve similar shapes or behaviors because they face similar pressures. Meanwhile, close relatives may become dramatically different after adapting to different environments. Synapomorphies help cut through that confusion. They are the inherited clues that suggest one set of organisms shares a more recent branch than another. A cladistic tree is not a direct photograph of the past. It is a hypothesis built from evidence. Scientists compare observable character states, anatomical structures, genetic sequences, developmental patterns, fossil features, and other biological details. From those observations, they infer which branching arrangement best explains the pattern. The common ancestor itself is often not directly observed. It is reconstructed as the most reasonable explanation for the shared traits found among descendants. This makes cladistics scientific rather than dogmatic. A proposed clade can be revised or rejected when better evidence appears. If a new fossil, a new DNA analysis, or a better interpretation shows that a group was built incorrectly, the tree must change. Cladistics works because it makes evolutionary assumptions visible and testable. One of the most important rules in cladistics is that descendants never leave their ancestral clade. In casual language, people often say one kind of organism evolved from another, as if the new form has escaped the old category. Cladistics treats that as misleading. If a lineage arises within a branch, it remains inside that branch, no matter how much it changes later. This is why some familiar words become surprising under strict cladistic logic. If fish is defined as the group containing the ancestors of land vertebrates, then all descendants of that ancestor must be included. That would mean amphibians, reptiles, birds, mammals, and humans are inside that larger fish branch. Everyday language may resist that conclusion, but the tree does not. Descent is permanent. This is where paraphyletic groups become a problem. A paraphyletic group includes a common ancestor and some descendants, but excludes others. Many traditional categories work this way because they were created from appearance, lifestyle, or convenience rather than complete ancestry. Older uses of the word reptiles often excluded birds, even though birds descend from within the reptile lineage. The word fish often excludes land vertebrates, even though land vertebrates came from fish-like ancestors. Worms can describe a body shape shared by many unrelated or only distantly related organisms. Cladistics calls these grades rather than true clades. A grade describes a level of organization or a general form. A clade describes a complete branch of descent. Those are very different things. When a group turns out to be paraphyletic, cladistics gives scientists two basic options. They can expand the group so it includes the missing descendants, or they can stop treating the old group as a formal clade. This can feel strange because it disrupts familiar words. But the purpose is not to make biology sound complicated. The purpose is to avoid classifications that tell a false evolutionary story. If birds are deeply nested within a larger reptile branch, then excluding birds from that branch hides part of the history. If humans descend from within a broader fish lineage, then pretending humans stand completely outside that ancestry creates a clean category at the cost of accuracy. Cladistics accepts the messiness because evolution itself branches from within older branches. Cladistic classification is deeply nested. Every branching point can define a clade, and every clade can contain smaller clades inside it. An organism can therefore belong to many clades at once, just as a person can belong to a family, an extended family, and a larger ancestral lineage. This nested structure is not an artificial naming trick. It reflects the way evolution works. New lineages do not appear outside the tree, they split from existing lineages. When a tree becomes more detailed, adding a new group may also add a new named clade and a new level on a branch. That can make classification look crowded, but it also gives scientists a precise language for describing relationships. The goal is not simplicity. The goal is ancestry-aware structure. Extinct organisms make the picture richer and more difficult. Fossils often preserve combinations of traits that do not fit neatly into living categories. A fossil species may look like it lies close to the line leading to a modern group, but cladistics usually treats extinct forms as side branches rather than confirmed direct ancestors. This is a careful choice. The fossil record is incomplete, and even a species that resembles an ancestor may actually be a close cousin of the true ancestor. By placing extinct organisms on branches, scientists avoid claiming more certainty than the evidence allows. These side branches are still extremely valuable. They reveal transitional patterns, show when important traits appeared, and help fill in the structure around living clades without pretending every fossil is a direct forebear. Stem groups are especially useful for understanding extinct relatives. A stem group includes lineages that are closer to a particular living clade than to its nearest living sister group, but that fall outside the modern crown group itself. In simpler terms, stem organisms are relatives on the approach to a familiar branch, not members of the fully defined living group. They help show how a set of defining features accumulated over time. Instead of imagining evolution as a sudden jump from one modern category to another, stem groups reveal stepwise change. Some extinct relatives may have one or two traits associated with a later clade, while lacking others. Cladistics gives scientists a way to place these forms accurately, without forcing them into categories designed around living species. Cladistics has created real tension with traditional taxonomy. Classic taxonomy uses ranks such as kingdom, phylum, class, order, family, genus, and species. Those ranks are familiar and useful, but they were not originally designed around modern evolutionary tree building. As cladistic studies improve, they can reveal that an established name does not match ancestry. A genus may need to be split. A family may need to be expanded. A long-standing group may turn out to exclude descendants that belong inside it. This can frustrate people who want stable names, but names are not neutral. They shape how biology is taught, compared, and understood. If a name suggests a natural evolutionary unit while hiding part of the branch, it can mislead. Cladistics pushes taxonomy toward evolutionary honesty. Today, Cladistics is the most widely used approach for classifying organisms because it aligns biological categories with descent. It does not make evolution perfectly tidy. Hybridization can blur boundaries among very close relatives. Fossils can be incomplete. Different evidence can support different tree arrangements until more data arrives. But the central principle remains powerful: classify life according to the pattern of common ancestry that best explains the evidence. Under that view, every organism is part of a branching, nested history. Familiar labels may need revision, and some everyday categories may turn out to be grades rather than clades. But the reward is a classification system that reflects life as it actually unfolded, not as a ladder of types, but as an enormous family tree of branches within branches. If you found this helpful, please like this video and subscribe for more explainers.